All right, let's get this show on the road. Welcome everyone to the June 2019 Soils Network of Knowledge webinar. I'd like to introduce Mark Farrell. Mark works with the CSIRO in Adelaide, and recently he led a GRDC funded project investigating the potential role of biological farming inputs in Australian broadacre agriculture. Mark's background is in biogeochemistry, and he's particularly interested in the interactions between organic carbon and nitrogen in soils and the importance these have in ecosystem functioning and agricultural productivity. He's studied many different land uses and ecosystems, both within Australia and internationally, and looked for the commonalities and differences of the processes and functioning between these two systems. So that's kind of why he's going to present today's webinar on understanding biological inputs, and one of the most requested topics we've had.
Hello everybody, good morning. Thanks Abby for giving me the opportunity to speak to you all today. I will try and rein myself in. I'm aware I can talk with a thick accent and rather too quickly. So I will try to keep myself quiet because, of course, through this medium, I don't have the sea of lost faces looking at me like I normally do when I realise I've gone off a bit too fast. So yes, as Abby says, I'm a research scientist at CSIRO, where I've worked for about 9 years. Worked on a range of topics, including plant nitrogen uptake, soil biology, biochar, compost, and carbon sequestration. And today I'm going to give a short overview of some GRDC funded research that we conducted between 2014 and 2017 on biological farming inputs. As with most efforts, this was a team sport. So I was the project leader along with my colleague Lee McDonald, also at CSIRO in Adelaide, Mike Wong, who at the time was at CSIRO in Perth, Mike Webb up at Townsville, and then Sasha Jenkins and Lynn Abbott at UWA, who were project partners. So the talk is going to be in 2 parts of roughly equal length. Firstly, I'm going to give a brief background to the topic as a whole. This was mostly literature based from the project, but one thing that we saw was conducting the lit review that formed part of the project was that there were lots of transferable things between different farming systems when it came to these amendments. And noting that the audience was going to be somewhat broader than just dryland grains, I felt that that would be helpful. And I'm going to spend a bit of time talking about the main findings from the research project itself. So one thing I really want to get through in this talk is that there are hundreds of different types of biological inputs, many different varieties, many different brands. They change quite regularly. So really, from a research perspective, we struggled with the concept of trying to focus on products themselves in too much detail. Instead, we took a step back and looked more at modes of action and how groups of products might behave. And taking that approach, we were then able to consider whether or not plausibly one or more of these amendments might work to address a specific soil constraint or other constraint to production. So the main modes of action, we've got direct nutrient value. Composts, manures, clearly you apply them not just for other factors, but because they contain nutrients. Not so much things like biostimulants that are classically defined as not having a direct nutrient value. And of course, that is where some of the questions come from about their efficacy, that if you're applying them at such low concentrations, how can they possibly have much of an impact? There are plant physiological mechanisms, and these may be induced by hormones and other bioactive compounds in biological amendments. Also, interactions between microbial inocular that may be applied. So biological mechanisms, again, may be direct or indirect. By direct, that would be the direct effect of a microbe that was specifically applied. Or indirect, say you're applying a compost and that alters the microbial community structure in some beneficial way. Chemical mechanisms, classically, obviously things like biochar and organic amendments have liming effects. So yeah, that's covering that category. And finally, physical mechanisms. Here, again, more the heavier organic amendments, compost, manures, that physically alter the structure of the soil. But that's notwithstanding the fact that you can get altered microbial communities, altered plant exudates that may, at least in the rhizosphere, alter the physical structure. So yeah, as you can see, there's those. We broke it down to those 5 main topics with a view that that would make it a bit more of a tractable issue than trying to look at 200 or 300 different products in any great level. So the next slides I'm going to go through, there are 2 or 3 that look pretty much the same. These are based on our review of the literature and basically trying to show over what time period different amendments might have an effect within each of the different modes of action. So first up is plant physiological mechanisms. And I'm going to go through this. I hope that people can see the cursor. I will move it very slowly. So on the y-axis is logarithmic, and that's to allow us to show whether or not it's a growing season, if it ends below a year, or whether or not it's 1 year, 2 years, 3 years, up to decadal or beyond. The width of the shape is the size of the impact, and the length is the duration, and the colour relates to the key on the right. So for plant physiological mechanisms, and bearing in mind this was done in the broad-acre context, clearly anything that works directly on a plant, and the plant is annual, can only last for a growing season, so that's less than a year. And you'll see that we would expect that something like a biostimulant, the point at which the greatest concentration of that chemical is in the soil is at the point of application, and it decreases over time. Whereas for something like a microbial community, you'd expect opportunity, if they do well, to grow before their influence wanes as the plant moves through its growth cycle. You wouldn't expect direct plant physiological mechanisms to be too high from things like manures and compost. That's not why they're applied typically, or indeed biochars or humates, although humates are mooted to have some biostimulant properties. So biological mechanisms, we've generally kept this as pretty tight, because there is, even for things like composts and manures, there is what few studies have actually really tried to unpick the impact of the amendment itself on the existing microbial community versus the impact of the microbial community that comes with the amendment, have been pretty inconclusive. So we did hedge on a narrower band here than you saw previously on the physiological mechanisms. But you'll see things like biochars, which we know mature in soil over time in their interactions with the mineral matrix. We might actually start seeing more impact over a longer term, and that's borne out in some of the more recent literature from work that's been going on for a number of years now, since the big flurry of biochar research back in 2010. Chemical mechanisms, so pH, buffering capacity and cation exchange. You wouldn't really expect biostimulant or a microbial inoculum to do much in that regard. This is ultimately fairly hard chemical impacts on the soil itself, much the same way that you'd apply lime or gypsum. So for products that are bulkier, you apply at higher concentrations, you'd expect a greater amount, but we know that manures and compost degrade over time and lose their influence. Whereas something like a biochar that's considered fairly inert, you'd expect to still be having a similar effect for quite a long period of time. So by way of summary to that literature review, and I should add that there will be links sent out to a few of these pieces of information at the end of the talk. By background summary, it was a highly diverse range of products, and the project itself looked more in depth at about 60 different compounds. We were tasked with looking at more compounds rather than looking more in depth at few compounds. With 60 different products, we didn't scratch the surface. So yeah, there are a hugely diverse range of products. But they are mechanistically categorizable, as I hope I've shown through that brief overview. Biostimulants, you're applying them primarily to impact on the crop that is growing that season. Logically, therefore, you would expect to have to apply them season after season to have an effect. Organic amendments, on the other hand, particularly things like biochars, you'd expect to last quite a bit longer, so you need to think about the economics of them in the context of 2, 3, 4 plus years rather than just the year at which you apply them. The most important thing to take away from this is expected mode of action, because if you understand the expected mode of action, you're there in a situation when you can look at what production constraint you're facing and how various different types of products might impact that. And this is a grab from one of the review documents that will be available. So I hope that gives a good background to where we were going with this project. I'm now going to move on to a couple of the highlights from the GRDC project itself. So as I said, we had a focus on breadth rather than depth. We examined over 60 products directly in this project. We were also able to focus to draw on a pretty wide range of amendments we had in our inventory from other projects in the past, such as those out of the Filling the Research Gap investigations into compost and manures, and also, of course, the biochar projects that were also led out of our labs here in Adelaide. So we focused on 60 plus some others, a lot of lab characterization, a glasshouse screening experiment, which was really just a bioassay. There wasn't really much focus on mechanism there. The main test was whether or not any of these were either standout, positive or negative. And then field experimentation. So across 8 different field trials, we looked at 38 different products. Now, we didn't have 38 different products at each site. Rather, we had, after we'd had various controls, we had 14 different products at each site, but together that made up 38 products across the whole 8 sites that carried out across the country. It wasn't all breadth rather than depth, though. We did have some mechanistic work that we were able to focus on. One of the recurring themes and wishes from consultation was that, were there products out there that gave better capture of existing soil nutrients? I can disappear down a wormhole talking why that might not be quite the right question I'd like to focus on, but an iteration of it was, if you have a legume in a crop rotation, how can the next cash crop make best use of the nitrogen that was fixed? And that was a question that we felt we'd be able to address quite succinctly in a targeted pot experiment, and that's what we did. And we also conducted a broader laboratory incubation looking at the impact of amendment chemistry on microbial function. So the 2 things that I will be talking about today in more depth are that range of field experiments and then the mechanistic study on nitrogen capture from litter. So as I said, we had 8 field trials. These ranged around the country. From Bunting over in WA, we had 3 in South Australia, Freeling/Jamestown, because unfortunately, one thing I can say is that no biological amendment makes a field trial fireproof. And the year our Freeling trial was due to be harvested 4 days after the Pinery fire, which totally razed it to the ground. So we moved next year up to Jamestown and had 1 year of experimentation up there. In Victoria, we took advantage of our collaboration with Southern Farming Systems, who ran the trial down at their Inverleigh site. Agroagronomy over in New South Wales, that's Barry Haskins, led the trials up at Rankin Springs and Parks. And then Northern Growers Alliance up in Queensland ran a site for us at Mount Tyson on the Darling Downs. Given time constraints, I'm going to focus more on yield in this talk, and only on the 2nd year, with the view that we were applying these products to the same plots year after year for the 2 years that we had of field trials, with a hope that we're able to see some sort of cumulative effect for those products that would have one. So I'm presenting here field data from 2016-2017. Yields under 100% district practice fertilizer as one of the controls range from 2.3 tonnes per hectare at Bunting to 7.6 tonnes per hectare at Inverleigh. That was the very wet year, the year that South Australia lost its electricity. So yes, we had a lot of rain that year, and it contrasted greatly to the year before, where a lot of the crops were clearly very water limited. So what I'm going to present now, rather than trying to show 8 different field trials relative to different controls, is I'm going to show the response ratio relative to the 100% district practice. So basically, if you see a bar that goes above this dotted line, you're looking at a significant positive effect relative to just having applied 100% district practice fertilizer at that site. Conversely, if it's below this line, it had a significantly negative effect relative to that. I should add here that none of these amendments were applied alone. They were applied on top of 50% district practice. We took the pragmatic approach that we weren't trying to completely change the farming system here. What we're looking at was how we could augment it. But if you're augmenting a system, that adds a cost, which, unless it creates greater profit, requires that the money for that cost needs to be found somewhere else. So reduced fertilizer rates seem the most pragmatic balance of that. And that was agreed between both the scientists and the grower group advisors who we were working with on the project. So first up is the alternative fertilizers. Now, I didn't cover these too much in the talks earlier because they sit a little bit outside what you'd call biological inputs, but they are fertilizers that focus on being softer on the soil, for want of a better terminology. Ultimately, you're looking at slow release or more balanced or focused in ways that at least the marketing would suggest are less impactful than throwing granular urea around. It's interesting to note that a couple of them, if you look at the error bar, don't cross over with this lower line. None of them are above, but most of them are around. And these were the ones that were, of course, we were most capable of looking at in the context of what is the nutrient load and matching it to the district practice. So whilst we can say that none of them were particularly damaging, none of them did particularly better, then instead of applying 50% district practice and 50% the alternate fertilizer, you got more or less the same result as applying 100% district practice. So nothing bad, nothing good. Biochars, likewise, we only use these at the 3 South Australian sites, and it was just one biochar, again, fell within the range. Biostimulants, not really much positive, and a lot of these tend more to the negative. That's not telling you that it had a negative effect relative to the 50% they sat on top of, but what it is telling you is that they didn't reach the same yield potential as what was achieved with 100% district practice. Humates, again, and there are a few of these that had really deleterious effects relative to the 100% district practice, and these would have fallen also as a negative against the 50%. So there was actually something negative happening here. Microbials, again, mostly tending towards the negative. Those two that are in the positive, look at the error bar and you would see it stretches a fair way below. There is no significant impact of any of these, but nonetheless, it is, they're not having a hugely positive or negative effect. The one type of amendment in 1 year that did was the organic amendments. And this was, whilst I won't, for various reasons, be able to talk about brand names for most of these, and I think I've already explained why I probably don't need to in the context of there being just so many different products, it is really difficult to make a clear case that this one is the product that will work for you. It's more this product type might work for you. But in the organics, this was a curious result. So this was a poultry litter at the 2 New South Wales sites. Chemically, it's not that much different than other manures that were applied either at the sites or at other sites, and it was the same poultry litter that was applied the year before at the same site. Yet this 1 year, we got this massive yield increase, well above 100% district practice, and well above the 200% district practice conventional fertilizer control as well. So in this 1 year, and it is pretty unsatisfactory that we can't explain why, we did get this absolute bumper growth, and it's probably due to a combination of things. but unfortunately none of the data that we have really pinpoints it. So that was the field experiments, and I do have a lot more data that I won't be able to show today on impacts on soil chemistry and soil biology, but I can talk about them in question time if that's of help to people. Moving on in the interest of time to the nitrogen uptake experiment, we did a pot experiment in the glasshouse here. We looked at 10 biostimulants and humates that specifically stated they would aid capture of soil-based nutrients. And as discussed in the prelude to this, we took that to be being better able to take up nitrogen that had been fixed by a previous legume crop. We had some isotopically labeled MEDIC litter available to us, which is why we used MEDIC. The same experiment could have been conducted with any other legume litter. We're focused on nitrogen availability, and this figure shows changes in nitrogen availability over time, looking at the soil solution. This is just total dissolved nitrogen on the y-axis and time on the x-axis. It's an excerpt of the whole data set. What's interesting is that at the peak point at which most of the nitrogen is being liberated after the legume litter was added to the soil on day 1. The litter control, so this is the one which had just the litter and no biostimulants, had a significantly higher amount of soluble nitrogen than any of the 5 liquids shown here, and indeed the 5 solid humate amendments also, which all clustered more around the no litter control. So there is something happening here showing that these amendments are reducing, either reducing breakdown of the litter to a soluble form, or enhancing plant capture of that litter such that we're not seeing it in a soil test. We also had a look at plant uptake. Now we grew these wheat plants in this pot trial up until the point at which the first pot reached anthesis. We stopped it at that stage. We didn't want to get to a situation where we had pot-bound wheat that was really being constrained more by the pot and the situation it was growing in, rather than the treatments that we're imposing on it. And the data I'm showing you in this figure is total nitrogen content of the plants, which is a good proxy for how much nitrogen was taken up. The reason I'm not showing this rather than capture of legume is that the shape of the figure is pretty much identical in both above and below ground. So this is the best integrator of the data that we have. And what you'll see is that for the most part, we have just these 2 liquid amendments that actually reduced uptake relative to the black bar, which is the litter control on its own. They're still well above the white bar, which was with no litter, but you'll see that there's clearly a lot of constraint here in these 2 products relative to the other 3, and that none of them actually are particularly enhanced over the products not being there at all, which is a bit of a curious and disappointing result. Now we have data still coming through the lab on microbial communities as affected by these amendments. But yeah, so that's ultimately what we found, which is pretty contrary to what we were expecting. So quickly going over the summary. There was a large variability in chemistry that I haven't shown, but what we did find in the literature was that predictions of mode of action are possible, even if those modes of action might not eventuate in your system. We did find across 8 trials in 2 years limited effect on grain yield, which is a little disappointing. A mechanistic study also indicated no positive effect of biostimulants on wheat nitrogen capture. Now I should emphasize on these 2 points, this is what we found in these studies. It is not an exhaustive project. Given the breadth of amendments, it would be very hard to have an exhaustive project. Broad recommendations. It's important to understand the constraints of production. Rather than going to a product, think about why you might be thinking about needing a product. Know that certain types of amendments are more likely to affect and address some constraints than others. I'd strongly encourage situation-based experimentation at the farm site, and this can be done with replicated field strips and yield monitoring or precision agriculture approaches. And we did produce a guide that was available, and I see has been handed as an electronic handout to at least provide some suggestions as to how that could be done in a way that will give you repeatable findings. And with that, I'd like to thank you for your attention and hand over to the chair for questions.
Thank you, Mark. That was great. And I like what you said about getting people to test or to look at stuff in their own situations, because as you said, it's very complex and difficult to have broad rules of thumb, if you like. So there's a few questions already. Someone's asked if they'd be able to access the PowerPoint, so I'll just put that out to Mark if he wants to put it in a PDF. I'm happy to distribute that. If people would like it, just get in contact with me through that soils.unit@dpi.nsw.gov.au or my own personal email, and I can get that off to you. Lisa Lobry de Bruin has asked if the graphs on slides 5, 6 and 7 were based on real results or hypothesized impacts.
So they are based on, well, they're based on, I hesitate to call it an expert panel, but I think that's what it's formally called. Ultimately, the authors of the review, which was the authorship team that I showed you at the start, we reviewed literature from about 300 or 400 different sources, and that was where, in a qualitative manner, where that led us to. So they are qualitative, not quantitative. There is data that guided our, the relative sizes and shapes of those bars, but no, I would not. They're not quantitative information.
Yep. Okay, great. Thank you for that clarification. Thanks for the question, Lisa. Vicky Brunn has asked what you mean by litter control.
So I'm assuming that was in the nitrogen experiment. Bear with me, I'll go back to that slide. Yeah, so we had, litter control was just the medic litter without any of the amendments, which is the zero, which is, sorry, I've used the wrong terminology on the figure. That would be why for the confusion. Yeah, so on the figure you can see the two bars at the far right hand side. You got the no litter control, which was just the soil, no amendment and no litter, and then the zero control, which had the litter but no biostimulant or humate. And then you've got the 5 green bars, which are the 5 biostimulants, and the 5 brown bars, which are the 5 humates.
Okay, I'll just clarify. Vicky's just put another comment. It was not very clear to me that any of the tests provided any real results. Can you say which method or what that method was that was best?
So I'm not quite sure how to answer that. I mean, this experiment here focused on whether or not, focused on answering a question that a lot of the products claim to address, that they assist in taking up nitrogen and other nutrients that are available in the soil, thereby reducing dependence on mineral fertilizer. In the context of that, the best test that we thought was, one, using isotopes to be able to see where the nitrogen had come from, whether or not it had come from the litter or the soil, which I've not presented here for brevity, but the shape of that figure is almost identical to this. That matters less in my mind than what the total amount of nitrogen was, which is what is presented here. And there was no positive effect of any of these amendments on improving capture of nitrogen relative to them not being there. And indeed, a couple of them, the first two, liquid A and liquid B, actually reduce that capture. I should also say that the above ground and below ground biomass figures are again a very similar shape and story to this one.
Okay, thank you. Hopefully that's if that's still unclear, Vicky, just get in contact with me and we can talk about that later. Julian White has a really good question. He said you talked about the quantity stuff around yield and things like that, but did you look at the qualitative stuff? I'm assuming it means product quality.
Sure. So we did look at grain nitrogen and phosphorus content. Obviously, grain nitrogen content is easily convertible to protein. Again, no significant findings, no systematic impacts beyond the logical impact of increasing amounts of fertilizer. We weren't able, given the project envelope, unfortunately, we weren't able to do more in-depth sort of plant quality studies and indeed grain quality studies. So we don't have things like harvest index, which might have been useful in the context of looking at longer term effects. And no, apart from a couple of the grow groups that did do some extra grain quality analysis, we weren't able to capture that data, I'm afraid.
Yeah, okay. Patrick Francis has said, could you explain what are the alternative fertilizers compared with organic fertilizers, please? I guess that's a terminology thing.
Yeah, no. So yeah, this included things like some of the, I think, what are termed balanced fertilizers that you might find at places like Landmark. I'm remiss to give, I don't really want to go down the line of giving product names. But yeah, the range that Landmark, for instance, market quite heavily. Also things like struvites. There were a couple of struvite-based fertilizers in that collection of red bars also. And they were applied to make up the difference between the shortfall of the 50% normal fertilizer and the 50% addition of them, because they were the one fertilizer type that we could, of course, apply in a very targeted manner relative to their nutrient content.
Okay, I hope that answers that question, Patrick. I know that Luke might have had a few questions.
So just one thing, Mark, the data you showed was mostly on grain yield. Can you describe what investigations you undertook to examine impacts on soil health?
Yeah, so we took soil samples, again constrained unfortunately by the size of the project relative to its budget. We weren't able to do as an in-depth job as perhaps we would have liked if we had just 1 or 2 sites. But across the whole 8 sites, we took soil samples between 6 and 8 weeks after sowing, trying to capture that point at which the wheat is really starting to take off. And in there, we looked at available nutrients, things like microbial biomass, carbon and nitrogen also, to give an indication of whether or not the microbial community size has been altered. And we also, for select treatments, had a lot of microbial community analysis work done on them. And the reason I haven't presented this data, one, is because you'd imagine across so many different treatments and sites, it becomes incredibly complex to try and distill down to a presentation. And 2, again, as with the grain, there was very little systematic impact. There were some significant differences between treatments and between each other, but there were very, very few treatments, and none of which you could really say were a positive or a negative in relation to the control treatments. So yeah, rather frustratingly inconclusive.
Yep, fair enough. So Mark, what do you think of the next steps for research into this space?
I would, it's a strange one for a research scientist whose response would normally be, give me more money and I'll do more. No, that's not the way forward with this. I think there are two big barriers to that. Firstly, results are so situation specific, and secondly, the sheer breadth of products that are out there. I don't doubt you could invest research to find out exactly what a specific product did, but I'm really not sure that's the role of GRDC's funding or indeed CSIRO or anybody else's research efforts. So there are two possible ways forward. There is, the problem with the industry as a whole is that it is highly fragmented, which makes I have a lot of sympathy for a lot of producers in these products, because they're in a situation where they can test it themselves, but they'll always be challenged that, well, it was your product, so of course you found that. that's an easy way to shoot it down, and I don't think that's always particularly fair. Or they can then try and engage somebody like CSIRO or DPI to do a rigorous experiment for them. First thing they'll come across is, of course, that that's a rather costly exercise. Secondly, you have the question of whether or not, well, it was still funded by them, so are we really finding the results? And thirdly, you've then got a strategic question as to whether or not that is really the work that CSIRO and DPIs and universities want to be doing. So what I think is a much better approach is that growers or advisors really take to looking at doing region-specific trials on types of amendments they think that would work, and doing this in a very basic way, using strip trials, or even better, replicated portions of a strip using precision agriculture equipment if that's available to them, and then just doing some very basic comparisons between what you would have done normally, what you've changed. There just isn't the breadth of products that are out there makes a study like the one that I've conducted, which was sort of stuck in between trying to get some mechanistic findings whilst having broad applicability. I don't think we really got there, to be honest, and I'm not entirely certain, even if you double the budget, that we would have done. It's a bit of an intractable problem because there are just so many different variables, and I think the best information that's going to be available to growers is to do a little bit of rather than just having. I know growers quite often will run a strip themselves and then eyeball it, but with yield monitoring, you can do a pretty good job of doing 2 or 3 strips, one or the other, randomized, and that's really not too different than what we do as a scientific trial.
Yeah, I think that's a good point. And I think the thing is to think really hard about what it is that you're trying to do in applying that amendment, and what therefore what we want to measure.
And that ultimately biases towards only having information on grain yield. It's not going to give you much on the longer term impacts of soil quality. But there's nothing to stop you doing a bit of focus soil testing before and after on these treatments as well, if that's what you were going to do, and build that into your soil testing program that you would have done normally. Yep,
that's right. Okay, we've got a few more questions now. Thanks for that, Mark. Dominic Osborne wanted to know if there was a difference in impact between incorporating or not incorporating the product into the soil. It's a good question.
It is a good question, and it is one that we unfortunately didn't address, given the number of products we already had. Our application method was on the basis of manufacturers' recommendations. So where they wanted it, when they wanted it in the row with the seed, that's where it went. When they wanted it surface applied, that's where it went. If they wanted it applied a couple of weeks prior to seeding, that's what we did. We didn't really have much scope to look at that. I would suggest that, given the research that's been being done on looking at adding particular organic amendments at depth along with deep ripping, that's definitely an avenue for future research. But no, we weren't able to cover that question.
Yeah, and I think there has been other work that's been done on placement and things like that of different kinds of, not the breadth of products that you looked at, but that other people might be interested in around that.
Yeah, and even placement of fertilizer itself in that regard.
Yeah.
Always at DPI.
That's right, the DPI Wagga stuff, and we've had Esan talk a little bit about what he's been doing in that regard. Richard Doherty says, do you feel there may have been an increase of yield if it wasn't a monospecies? And then he said, like cover cropping.
I honestly can't say. We did. It ended up being pretty much entirely wheat. We did have barley at Freeling, unfortunately. That wasn't. I mean, ultimately, that wasn't the focus of the project. And I should add here that we took a lens, being not just we took a lens. The remit we were given by GRDC was investigating how these products will fit within existing systems rather than looking at broader whole-scale changes. I do have a personal interest in cover cropping and working on a project funded under the last NLP round in that space, and it's something that's at the back of my mind, but I don't have any data to either say one way or the other on that one, I'm afraid.
Okay, thank you. Bede O'Mara's curious about how you chose the products and how they were shortlisted, what the process was around that.
Okay, this was a convoluted process. One I've tried to wipe from my memory. It was there were a number of different avenues. Primarily, as I mentioned, but haven't actually explicitly stated, we had several grower groups who were working with us on this project. So you had Libby Group in WA, Sanfa over here in South Australia, Southern Farming Systems, Agroagronomy, and Northern Growers Alliance. Now, we asked them to use their networks to find out which products and which types of products were of most interest to their members, collaborators, and clients. From that, we then went down the approach mostly of me and the other scientists being upfront with producers of these products as to what we were doing, what we wanted to do, and whether or not they were happy to give us something to be involved. Most said yes, some said no. Mostly we left that where it was, but there were a few key ones that fell in that no category that we had multiple grower groups saying, look, we really want you to look at this, at which point they, like any other member of the public, went and sourced the product. But given the fact that we're working under we would, and a step further back, CSIRO wouldn't have taken this project on at all if naming names was on the cards. GRDC were quite happy with that, and I appreciate that has been a frustration to some of the listeners at times. But that is, you'll appreciate there's significant legal ramifications one way or the other for either promoting or negative conversations about products. And we went, having taken that early step earlier on to go, well, look, let's think more about them in terms of their modes of action. We felt that we got around that need to some regard. I'll be upfront, I can't say there isn't a wonder product out there that we didn't test that worked perfectly. I don't know that. I don't have any evidence to refute that or to endorse it. But yeah, ultimately, most suppliers were pretty keen to see their products involved. Some, like I say, less so. But yeah, our product choice primarily was driven by the grower groups.
Okay, thank you. Yes, that's always difficult. We find that difficult at DPI about appearing to endorse or not endorse a product, being a government agency, as you say. Yeah. It's fraught.
Yeah, and it rapid, and you're on a hiding to nothing, because in this context there is, yeah, formulations. I would reckon already 2, 3 years on, that a good number of the products that we tested there aren't available in that form any longer.
Yeah. Okay. Oh, so further to that,
without us having provided endorsement or otherwise. So
yeah. So they've maybe learned something. Well, Bede's just said further to that, that's great to that various modes of action were screened rather than naming specific products. So that's nice to hear. Vicky Brunn has another question about the graph on N uptake. She's asked just what is the solid material as opposed to the liquid? Are the liquids manures or soil conditioners or?
So the liquids were 5 branded biostimulants that were available in liquid form, and they were applied at field rates to the pots. The solids were humates, which can be classed as biostimulants and are under some definitions, but they were primarily solids. So yeah, ultimately, just by nature of what we had in the inventory, all our humates were solid and all our biostimulants were liquid.
Yeah. And did you define them as that? Because there's not really any legislation or anything around this, so people can call something anything really in this fora, can't they?
Yeah, there are. Whilst I'm not sure on I don't have a cast iron answer on the legislation. There are certainly recognised definitions coming out of a lot of the very in-depth research being conducted in Europe on this. Humates, the humates here were all derived from coal extracts of one description or another. Whereas the biostimulants, their source was a lot more mysterious, shall we say. They may well have included some lignite extract as part of those products, I don't know, but they were marketed as a biostimulant, whereas most of the humates were marketed as humates with biostimulant properties.
Okay. I just also draw people's attention, if they're interested in that, there is actually a publication that was done a few years ago by DPI on humic products that goes into a lot of what that stuff is and how that, whether they've got potential within agriculture, by a woman called Kim Billingham. It's I think it's available digitally. Could Mark put up the slide which has the check sheet of what resources looking at achieving was near the beginning? That's from Richard Doherty.
So that figure is in Lynn McDonald's summary review
Publication.
Again, as per just to follow up on that earlier question about how quantitative those initial bar charts that we put up were. Again, this is what we have drawn from a pretty in-depth review of the best information that was available to us at the time of writing 2 years ago. But that's why there are grey squares and white squares rather than numbers of rankings. The information just out there to make those quantitative statements.
All right. I think that's all. In that case, I would like to thank Mark very much. It was really interesting, and I hope that everyone listening got as much out of it as I did. And we thank you for your attendance. Yeah, thanks very much, Mark. It was great.
Not a problem. Thanks for having me.
Thank you for listening to this Soils Network of Knowledge webinar. Remember, New South Wales DPI also facilitates other community of interest products around soils, such as our Twitter account and the quarterly newsletter All the Dirt. If you'd like to access these, you can use the information that's now displayed on your screen. And don't forget, you can also subscribe to the webinars so you never miss out. You will receive a monthly invitation, and the choice to attend is up to you. If you've missed a past webinar, you can catch up by looking at the transcribed recordings on the New South Wales DPI Agriculture YouTube channel, or the Go-To stage, which has the recordings that are unedited, and those links are also on your screen now. Thank you very much for your interest in SNoK, and we hope to see you online in future.